Good evening, friends. This is Dr. Rajkumar. Again, welcoming you on behalf of IPS Vadodara and Bombay Psychiatric Society for yet another session of Brain on Wednesdays. To chair today's session is Dr. Sarika Dakshikar. She is an associate professor and head of the unit at the Department of Psychiatry in Grant Medical College and JJ Hospital. Has 20 years of experience in clinical practice. Areas of interest are addiction psychiatry, general addiction psychiatry, and consultation lies in psychiatry. The topic for the evening is Dr. Sarika, you can take over.
Thank you, sir.
Unmute.
The topic for the evening is psychoimmunoneurology, and we know it's a complex thing, and complex in a sense that one thing is related to the other, and most of our patients have multiple complaints, and we keep thinking ki, "Arey ye bhi hua, is it coexisting? Is it different entity?" But there is a connection, and that connection will be unveiled by Dr. Malay Dave. Now, most of us know Dr. Malay Dave, and Malay, for those who have met him in person, they already know he is very humble and he's very modest in sending his CV. I'm only going to, I mean, he wrote only a few points in that. Uh, he's consultant psychiatrist in a private practice. He's PG teacher for last 12 years. He has won the Bombay Psychiatric Society Silver Jubilee Prize at ANSIPS Bhubaneswar and Anil Avisha Best Paper Award. He has 12 publications to his credit. He has been on honorable president of Bombay Psychiatric Society. He's currently member of he's member of IPS UG Education Subcommittee, and he's advisor for IPS Biological Psychiatry. The areas of interest are neuroscience and genetics. So I think most of the conferences that we attend, we have at least one Dr. Malay Dave's lecture either on neurobiology or on genetics, and we know how complex things appear when we try to read out themselves. But he actually makes it very lucid for us to understand, and things start making sense suddenly. So without wasting much of a time, over to you, Dr. Dave.
Yeah,
You are
mute.
muted.
Okay, good evening, everyone. Thank you, Dr. Rajkumar, sir. Thanks, Dr. Shobha Nair. Thank you, Sarika, for those kind words. And today, again, we are going to look into something which is more basic, and we need to kind of keep in mind when we are trying to understand psychiatric disorders. So, today we are going to look at neuroimmunology, or the older term which was there was psychoneuroimmunology. Actually, these are four terms. One is the psycho part, one is the neuro part, one is the endocrine part, which we are not looking into today, maybe another time, and the immune part. So what it basically means is that we are going to look at the immune function, how the nervous system kind of interacts with the immune functions, and how does it produce neuropsychiatric symptoms. This is what it is meant. We need to understand that psychoneuroimmunology or neuroimmunology or endocrine immunology or NEI, neuroendocrine immunology, these are subspecialties in psychiatry. So a lot of basic research has been done into this area. We have got a lot of information, but even before I start, let me give you the caveat that we do have a lot of information. We do have lots of findings. Most of them are in the domain of association and not strictly causation. But nonetheless, as science advances in terms of our understanding of basics, the introduction of newer concepts and newer technologies, it makes us, it gives us a lot of things that previously we might not have thought important, and which now have the potential of becoming biomarkers, which we are always in search for to kind of clinically be certain as to what we need to focus on, how a patient is going to improve, and so on and so forth. Now, before I start, we have previously talked about epigenetics. We have talked of epigenetics was last time. We talked of something about stress sensitization last time when we talked of FKBP5 gene epigenetic issues with that, and how a person is sensitized to stressors and its effects over the lifespan. Now, this is going to add one more angle to looking at things. And as Sarika mentioned, this is one area which kind of integrates a lot of things that goes on inside a patient's body. This will also make us wonder whether psychiatric illnesses are, so to say, do they have origins in the brain or the nervous system itself, or they start somewhere and the final symptoms that we see as presentations from our patients, which are brain derived or brain related or disorders of the nervous function. So what we are going to look, I'll just share my presentation, and yes, is it visible? Okay. So psychoneuroimmunology, or PNI as it is called, or basically now you can just focus on the last two alphabets, that is NI, that is neuroimmunology. Now, what we are going to look at, even though the index is quite a bit, I'll try to make it as easy to understand and see what we can remember in all these things. So this is what we are going to look at. Okay, now this first couple of slides are basically the take-home messages from this presentation of today. And yes, now we understand that when we look at psychiatric illnesses and non-communicable diseases in general, at the base, in the background of most of the illnesses that kind of afflict modern humanity, there is this chronic inflammation. Chronic inflammation can be a cause of multitude of things. One of which is T cell dysfunction, which is again immunological, and other things. But this again are not really independent. They are integrated, or rather work in an integrated fashion, and cause a lot of illnesses in terms of diabetes, cancers, cardiovascular disease, and depression. This four being in the top 5 of the most problematic non-communicable diseases in every decade. The WHO gives a list of the morbidity and mortality which is associated in terms of a descending order, and in top 5 we are going to have this 4. We have not mentioned here degenerative diseases like dementia, but well, if that is there, then this 5 are usually the top 5 causes. What are the functions of the immune system? Now, functions of the immune system, first 2 are what we are aware of, what we have studied in our MBBS days, what we are looking, when this first 2 are being discussed, are always discussed when we interact with our colleagues from other medical specialties. But apart from that, what is our interest are the last 3. One is survey, control, and maintaining the gut microbiome. Now, gut microbiome, we know, again, we'll probably do that later on. Gut microbiome in short means all the organisms that are resident in the gut. Would be bacteria, viruses, helminths, fungi. They form the microbiome, the sum total of all the microbes in the gut. Now, they have to be maintained. It's like, we have a nice lawn, and if we allow that lawn to grow, we will have more weeds than the lawn. And when something like this happens, the gut microbiome is in a stage of dysbiosis, and it can predispose an individual to a number of illnesses. The starting point of many illnesses is in the gut, not because of what we eat, but because of the microbiome function of the gut. So most of the gut microbes are commensals, means they stay with us and they work with us, and they are in a kind of a balance. And this commensal nature of this microbiome can change into a parasitic form if the growth is unchecked, if the functioning of certain species of microbes in the gut is unchecked. Now, to keep a check on that, because microbiomes then, when they are not in a commensal state, they will become pathogens. And these pathogenic signals will be picked up by the immune system and the weeding out will be done. Another important thing from our perspective is sculpting of brain circuitry. Now, the commonest understanding of immunity is, well, fight against pathogens, that is one. Now, later we understood that even cancers are immunogenic in nature, so to remove cancer cells, that is two. But what is also important is that immune processes are like scissors, molecular scissors. They will work on brain circuitry. Brain circuitry will always need fine tuning. Synapses that are dead, not functioning, problematic will need to be removed. Cells that are worn out, fibers that are worn out will need to be removed and repaired. Now, all this is fine tuning of the brain circuits. So this one concept we looked at when we discussed the connectomics. Brain circuitry, sculpting of brain circuitry is immunogenic. So immunity is not only infection related or cell removal related, it is kind of designing, editing one gut microbiome, second, the connectomics of the brain, and thirdly, and most importantly, it regulates neural development and neural communication. So right from day one, the same immunological processes that in adulthood, or as we understand, would fight pathogens or fight cancer cells, are the same processes in utero have helped in neural development and neural communication. Okay. Now, those were the take-home messages. Now, what do we understand and how do we kind of look at the immune system? So, first of all, we need to understand that there are certain lines of defense. Means anything that our body encounters, which is not our body, which is foreign or alien, will have to be processed. So what are these lines of defense? One is the physicochemical barriers. Now, immunity is basically of two types, innate and acquired. Innate is what we are born with, whatever is the current state of our body systems. Okay, skin and mucous membranes, these are the first points of contact. When a pathogen or whatever is encountered by this, it sets off a chain of reactions which we call as innate immunity. Acquired immunity is more to do with cutaneous and mucosal. It's basically the same thing, but this is developed over a period of time. It stays for a longer period of time because this is the immune system that learns. This is fast action, doesn't last for long, taken over by this, which will last a long term. And we know that certain illnesses, we all got vaccinated and we developed acquired immunity. So now for those illnesses for which we were vaccinated as children, we are immune to that. Different types of cells are involved in each of this. For the innate, we have phagocytes, that is macrophages, neutrophils, and natural killer cells. And for the acquired, we have lymphocytes, B lymphocytes and T lymphocytes. We also have for innate macrophage related cytokines, IL-1, 6, tumor necrosis factor, and interferon alpha. And for this, we have the other ones, interleukins 2, 4, 10, and interferon G, gamma. So innate are trained. So they will do their work. But as I said, it is short term. But when it comes to long-term memory, so even if I have been exposed to something in a childhood and not later exposed, and I get exposed to that same organism or same kind of an insult 50 years down the line, there is going to be a memory of the same. Okay, and innate, the other molecules are the complements and acute phase reactants. We'll get to that in a bit. Whereas acquired is antibodies of different types. So this is how it works. This is the first contact where the innate immunity comes into play, and then it is adaptive immunity which takes over after this, and then over a period of time, that is second exposure. So it is pretty clear that whatever viral or bacterial attack, in few hours, all these macrophage, neutrophils, they natural killer cells, it's their work to kind of look at the threat, neutralize the threat. Then this leads to, this activates, this interacts with the first exposure of adaptive immunity, where we have the different lymphocytes. So there will be development of antibodies or marks, and that will take care of this invasion. And later on, even if there's a little infection, we are ready. So we remember. So there is the memory part of this. This is what we have looked at. So what does the innate system do? Innate immune system, basically surveillance. Mononuclear phagocytes or macrophages and natural killer cells will constantly work as sentries or guards and keep a check of what organisms or foreign stuff, non-self stuff is there in contact with the body, whether it is the surface or the mucous membranes, or if there is some injury, there's a penetrating injury and the organisms invade that. So their duty first, the innate system is surveillance. So surveillance. So we have this neutrophils, macrophages. Okay. We have seen this monocytes, right? And after the threat is processed, okay, whatever was the attack now develops an antigenic response. So some representation of the organism is now present on, or rather it is it stimulates the development of certain marks or receptor like structures on these cells, all these three. So now next time also they are ready that okay, we already have some memory, little bit of memory. This is not a very long term memory which is important for the development of the acquired response. There are different types of cells. When we look at the different kinds of cells that are involved in both types of immunity, acquired or innate, they all develop from hematopoietic stem cells, the myeloid progenitor and the lymphoid progenitor. There are two cell lines which develop from the stem cells. The myeloid ones, they finally lead to what we see in the blood. So we have erythrocytes, platelets, and different types of white blood cells. So neutrophils, monocytes, eosinophil, basophil, mast cell, and we have the dendritic cells which are seen in various tissues. The lymphoid progenitor, on the other hand, forms T cells and B cells, along with natural killer cells. So natural killer cells, which are a part of the innate immunity, come from the lymphoid line, and all those monocytes and macrophages, they come from the myeloid side. This is how they develop over a period of time. So we have mononuclear phagocytic cells. So as a part of the immunity, the first thing is that they surveil, they keep a check, they work as security guards. And when a threat is detected, various types of mononuclear phagocytic cells get activated. Monocytes themselves, macrophages, microglia in the brain, which we'll have to look at in a bit of detail later on, dendritic cells, and reticular cells. Reticular cells are seen in lymphoid tissue, so spleen, lymph nodes, lymphoid organs. And this system, this reticular system, or especially the spleen and lymph nodes, represents a cross between the innate and adaptive, because the cells, this kind of cells, they originate from there, and even the B and T cell lymphocytes originate from there. So if a message needs to go from the innate to the acquired immunity, this is the crossing over where it happens. Natural killer cells we saw originate from the lymphoblastic heritage. What do they do? again, we are on the innate immunity. So viral infections, they'll take care of, but they also will detect neoplastically transformed host cells. So the body has a ability to also detect cells which are kind of growing faster and being cancerogenic, or rather would become cancerogenic at very early stages. It happens in all of us. So they are also perceived as a threat by the natural killer cells and get eliminated, especially hematopoietic origin, because natural killer cells develop from the lymphoblastic heritage, from the lymphoblast, early lymphoblasts. So natural killer cells, two things: viruses, they'll work on viruses, and they'll work on neoplastically transformed host cells. Because what happens is, this transformed host cells on their surface, their antigenic presentations will change, which are picked up, and then this will work. Second thing is immune signaling. So after they do the surveillance, threat is detected, those cells, natural killer cells and macrophages, they get activated, then there is immune signaling. Now what happens is, when there is immune signaling, when the cells get activated, they start expressing toll-like receptors, which can pick up viruses and bacteria. So this is a kind of an antenna which will kind of interact with the viruses and bacterial surface antigens, and different kinds of molecular patterns get activated. We have PAMPs, that is pathogen associated molecular patterns. What happens is, when these cells come in contact with viruses and bacteria, within the cell itself, there'll be a chemical change, there'll be a lot of chemical reactions and patterns, which the final aim of which is to neutralize the threat. So the pattern of chemical activation is called as pathogen associated molecular pattern, PAMS. Now, when it comes to, let's say, wear and tear of cells, so cells have died, body cells that have kind of worked and died. So when the cell dies, different cellular products, including DNA and other debris or neoplastic cell, if the growth is gone overboard, they get exposed to these cells, natural killer and macrophages, and a different kind of pattern is generated in these cells, which are called as the DAMPs, so danger associated molecular patterns. So a reaction could be a PAMP, which is for pathogens, or a DAMP, which is for DNA, other debris of dying or neoplastic cells. And this is actual inflammation, hot inflammation. This is called a sterile inflammation. All psychiatric disorders that we know of, especially degenerative disorders, okay, will fall into the gamut of sterile inflammation. So let's, for example, say there is a degenerative neurological disorder like a dementia going on. Cells are dying at a faster rate. Their products are exposed, damps are formed, and there'll be more kind of destruction of cells. So that process is in a positive feedback loop, but this is called a sterile inflammation. Intracellular signaling cascades in terms of activation of mitogen activated protein kinases. This is one thing which will start a signaling cascade. Again, nuclear factor kappa B. These are the intracellular signaling cascades. Then we have inflammasomes. What are these? These are cytosolic protein complexes in the myeloid cells that respond to microbial and sterile inflammatory stimuli. So in the cell, what will happen is, whenever the cells form a PAMP or a DAM, when there is an activation of signaling cascades, they will assemble all these chemicals inside the cell, will assemble to form an inflammasome. So inflammasome is a kind of how we would call it, if the closest analogy I could think of was a SWAT team, specialized team of few people who will go invade enemy territory and get things done. So, and then they are not in single units. There's a group of people. So it's a SWAT team. This is an inflammasome. So a threat activates. See, normally we have all these cells. We have natural killer cells. We have got all those macrophages and all those cells which are in a silent state. They are monitoring. All this is happening silently. So only when these processes are going on fast or too much, that is the time when we will start experiencing symptoms or what we call as illness behavior. So we may not feel okay because all these cytokines and all will be functioning more, and all these cytokines, which we'll see later, okay, will produce certain physical equivalents which are symptoms, and we would give it a label. We'll say malaise, fever, or whatever. One of the clinical important features or clinically relevant things that we need to kind of understand is that in obesity there is chronic innate immune activation, because the size and the number of fat cells is very high and fat deposits keep on happening. So it's basically an overload, activity overload on the fat cells. Now, whenever there is so adipocytes are overworking. So whenever there is any cell is overworking, all processes to maintain that overactivity will also be overworking. Whenever cells or systems are overworking, the wear and tear is very high. Now, the systems which handle wear and tear will also need to work, or rather overwork. So what happens is there is a chronic innate immune activation in obesity. So physicians will tell us that even though a person is overweight and nothing is happening in terms of symptoms, or nothing much is seen on investigations except the weighing scale, which is showing more weight than normal, there is chronic innate immune activation going on. And the best strategy is to keep it at chronic innate immune activation below a certain threshold, because these adipocytes are going to die. When cells die, all their products, all their products, that is DNA or cellular organelles, whatever that is, are going to activate dams. That is the danger associated molecular patterns, and it is going to keep the inflammation alive. So this is basically a diagrammatic representation. So we have, let's say, stress, external stress. External stress can be whatever, but it can put a certain pressure. How? We'll see it later. Can make people vulnerable to infections or can cause metabolic changes. So we have MAMPs and DAMPs. Okay. So these two we talked about here. Molecular patterns. So basically PAMs. Sorry, there's a mistake here. It should be pattern associated, and this is damage associated. This pattern presentation will cause a lot of changes inside the cell. This is a monocyte. So caspase is NLRP3 is a inflammasome. And this will again cause a lot of changes, which through the nervous route. Now, if this is happening in the gut, we have the gut-brain axis through the vagus. So all these signals are carried to the brain, and they will also, there are certain cells, the microglia also function as we saw areas of first contact, innate immunity. They will also get activated. Normally, they are not activated, but under the influence of this, and especially if this interleukins, when they cross the blood-brain barrier, they will also get activated, and then we'll see the whole gamut of things starting from here to here. Then we have acute phase response protein. So till now we have looked at two types of immunity, innate, acquired. Then we looked at the cells. We looked at the macrophages. We looked at natural killer cells. What they do the surveillance. They identify the threat. They set into motion changes which will take care of the threat, which can also become pathological and produce certain diseases. Another component is the acute phase proteins. Acute phase, as the name suggests, are acute, synthesized primarily in the liver, and their work is limit tissue damage, isolate and destroy invading pathogens. and set repair functions in motion. Now, these compounds or these chemicals or these molecules, we are a little bit more familiar with. So there are negative acute phase proteins, means the production of these acute phase proteins, the negative one should come down and make way for the positive. So there is obviously going to be a balance between negative acute phase proteins and positive acute phase proteins. But if we want a challenge to be taken care of, we need to have more acute phase response proteins. So what are the negative ones? Albumin, transferrin, retinol binding protein. Now, if they are, these are measurable. Albumin is measurable, transferrin is measurable, and they become a marker for chronic inflammation. So basically, this transferrin and albumin, they are those molecules which in a pathology report we do not pay much attention to, because we are not able to figure out what is the importance of that. Again, a solitary reading may not be important, but in the context of the whole report, yes. And we have the positive ones which will drive inflammation. So we have protease inhibitors, alpha-1 antitrypsin or an alpha-1 anti-chymotrypsin, coagulation proteins like fibrinogen, prothrombin, complements C2345, transport proteins like heptoglobin, and inflammatory proteins like CRP. CRP is measurable. It is a very simple test. It comes in all reports. But what is it? It's a marker. So if it is very high, means pro-inflammation is going on. Now we will wonder why it is happening. There is no fever or nothing. But yes, if a person is obese, the adipocytes will drive the inflammation process. So we'll have an increased CRP. Autoimmune disorders, well, increased CRP. Psychiatric disorders, in some CRP. So this becomes a measurable marker. The CRP is a clinical marker of ongoing inflammation. So complements, even complement assessment. Complement we always hear in terms of coagulation and things like that. But what do complements do? Complements are like postage stamps. So, we'll see in the diagram that whenever a challenge, a bacterial or viral or any antigenic challenge is dealt with, it is given a complement tag. So let's say C4 tag. Okay. Why does it give a tag? So that further of these, when they come up presenting that tag, they will be taken care of by phagocytic cells. So let's say C4a complement. What is it important from? It is expressed on synapses. Now, depending on the synapses, wherever it is seen, the microglia will go and phagocytose that. This is very important in terms of pruning. At certain points in our lives, pruning is very important. Otherwise, newer synapses will not form.
Sir, one
Now,
sec. Requesting organizers to mute the phone. I think it's Xiaomi. Yeah, thanks.
okay. Now, if there is an excessive expression of complement 4a, means what will happen? Even synapses that do not need pruning will keep on getting damaged or pruned. Now, this excessive pruning is supposed to be an etiologic role in some cases of autism and schizophrenia. Not in some cases, in most cases of autism and schizophrenia, but in variable amounts. Coming to the second component, adaptive immunity. So we have T and B lymphocytes. So again, like the innate immunity, there's a recognition phase. And what do these T and B lymphocytes pick up? They have receptors for the antigen. And with a repeat challenge, so suppose a bacteria has got a label of, let's say, positive, plus on itself. The next time when that plus is seen by a B or a T cell, only that particular B or T cell which has identified that plus sign before, they will get activated. So they work as clones. So this particular B or T cell will, when they divide, when we have more or newer B or T cells, they'll express that same thing. So they form clones. And a family of lymphotypes with identical antigen is called as a clonal line. So in childhood, I was exposed to an infection. I have a particular presentation on a B or a T cell. Those cells will divide because cells will also die. So the newer cells from that particular B or T will express the same antigen, say a receptor for the same antigen, and this becomes a clonal line. So it's like our family lines. It's like a surname, whatever we want to look at it. And the way of understanding this clonal line. So when we study them, we study this surface protein expression, and then we label them as CDs, or cluster of differentiation. So we have got CD1, CD2, CD25, CD15, CD100, and so on. So T and B lymphocytes recognize, they activate. What do they activate? They activate the proliferation and mobilization of the immune cells by means of cytokines. So they will activate various cytokines. We'll see that whole list. And what are the things that they do? So when it comes to B lymphocytes, they produce antibodies. And when it comes to T cells, they produce various kinds of cellular activity, which is going to finally neutralize the challenge. So both antibody mediated and cell mediated immunity is what we are talking about. So again, a simple representation. So these are the lymphocytes, B cells, different types of antibodies, different types of microbes. Now this, whatever is the challenge, and if this has been successfully eliminated, okay, so this is going to form memory. This is the basis of long-term innate memory, right? So when an antigen is bound to the antibody, this complex will be taken up by macrophage and finished. This is going to lead to more children of that same B cell. So this is a clonal line. And this pattern is always going to be expressed in terms of memory B cells for future use. So like how our brain functions, we do something, we learn something, we understand experience, we label experience, and it is stored in memory. The same thing happens at a cellular level here in B cells, which are dealing with antibodies or the humoral immunity or humoral acquired immunity. That is, there is a process of learning. So this is going to be remembered forever. Okay. Now, B cells, they kill pathogens in extracellular space. But if the pathogens have invaded a tissue, okay, then you need to have cell-mediated immunity, okay, which is taken over by T cells. So again, it's the same thing. So here we have an antigen and an antibody interaction. When it comes to T cell, we have another group of chemicals or molecules that come into picture, which is the major histocompatibility complex, MHC. This MHC is used for a variety of typing, similar cells, different processes in different pathologies. Okay. So when we have a healthy cell, it has got a certain representation of MHC, let's say class 1. Now, when that healthy cell is infected by something, it also expresses another one. Okay. So this presentation is called as an epitope. So normal plus infected will be MHC1 plus, let's say, MHC2. Now, this is a signal for the T cells to understand that, oh, something is going wrong. This MHC1 part is okay, but this MHC2 part is something problematic. So they will attack this cell which is infected and kill it. So this is a kind of, again, identity card which is presented by the cells, to differentiate between self and non-self. So cell-mediated immunity protects against viral and intracellular bacterial infection and to some extent tumor cells. The T cells also secrete interferon gamma. So it's not only that they are going to engulf and just kill it physically, but also secrete cytokines like interferon gamma that enhances the antimicrobial function of macrophages, including microglia. T cells of different clones, cluster designation or cluster of differentiation, this 4 plus and 25 plus are regulatory T cells. So here the war is going on, but then somebody has to call a truce. Okay, now enough of them have been killed. Now the more you kill, normal cells will get infected. And that is where this CD4 plus and 25 plus, which are regulatory T cells, will come into the picture. So in this, the antibodies, I had mentioned before that the place where, or the regions where, or the tissues where the innate and the acquired immunity get kind of come in contact with each other, exchange messages, is, let's say, the spleen and the reticuloendothelial system. And the interaction is chemical in terms of the use of cytokines. An endless list of cytokines, different types: interleukin 6, 2, 4, granulocyte, monocyte, interferon alpha, gamma, so many of them are there. This is just a descriptive list. Okay. Now, this is how the basic immune system functions. Till this time, we have not touched the brain much. At the most, we have come close is the adipocyte tissue, chronic inflammation, damp, maintenance of that chronic low level inflammation, obesity, and obesity related non-communicable diseases and complications. So we are still now in the periphery. We have not entered the nervous system much, and we have not definitely entered the brain. Our interest, apart from understanding the basic science, is how this brain interacts, or brain function interacts, with the immune system. So the brain has resident immune cells called microglia, which are there in the parenchyma. So microglia are there wherever there are neurons. They are resident, so they are going to be in place. Whereas trafficking immune cells will be those cells which will move, which will have the membrane representation of various antibodies, that is B cell, those kind of things will be in various other places, including the meninges and choroid plexus. So resident and trafficking, two types of immune cells. Now, under usual circumstances, the direct communication between blood and CNS can happen only at two places: the blood-brain barrier, that is what we know of, and second thing is the brain lymphatic system. That is a lymphatic system of the brain. There is a movement of fluid and small molecules between the CSF and the interstitial fluid. So this will be more central, wherever there is the interaction of CSF with brain tissue. Blood-brain interchange is everywhere, wherever blood vessels come in contact with brain tissue. Now, these two present possible route for cytokines from the meningeal compartment to the brain, especially the lymphatic system. And so, whatever is happening at the periphery, given a chance, if let's say the blood-brain barrier is not holding up, it is damaged because of an illness or degeneration, then all those peripheral cytokines, whatever we talked about, can all enter, especially the cytokines and traffic cells can enter through the blood-brain barrier and cause changes in CNS function. Okay, so this is a diagrammatic representation. So we have got a T cell and B cell. So these are basically the trafficking cells, and in the parenchyma, we will have the astrocytes here. So this is the resident one. So again, it's the same kind of a thing. Microglia is more resident. Okay. Now, when we're looking at the brain, we need to look at the macrophages in the brain, because again, it's the equivalent of the innate immunity. So they develop from the yolk sac, okay, migrate from the yolk sac to the brain. Like all neurons and all the cells of the nervous system, they originate from the yolk sac, multiply, move, and go to their final position. So early macrophage precursors in the brain become microglia, and those in the periphery become macrophages. This is the difference. Functioning is the same, but their regional location is different. So what do they do? Now, they are also nerve cells. So they are going to kind of form synaptic integrations with the dendritic spine. So it's not synapses between two neurons, but even microglia synapse. So because of that microglial activity, even the synapse gets affected. So what does microglia do? Like macrophages, phagocytosis, so debris, pathogen, foreign material, etc. The same thing. Okay. But since they are there in the brain, so here what they've tried to show is a microglia forming a synapse with a synapse, let's say here. And here it is going to edit the synapse. So that is pruning. Okay. So this is very important in refining circuits, developing an efficient connectome. Whatever is necessary stays, whatever is there is kind of tuned, fine-tuned so that it works very well. And whatever is not needed, okay, that is pruned. Now, this is a normal process. So as we looked at what are the functions of the immune system, one was, pathogen, okay, detect pathogens wherever they are, if it's an invasion or if they're resident in the gut or if the cells have become abnormal. But in the brain, what is going to happen in addition to all these things is the blood-brain barrier actually provides a good kind of a barrier for all the peripheral infections to invade the brain. So if I have got some UTI, I might not develop meningitis. So that infection is localized in the periphery. The brain is protected. But within that, what are the challenges? One is how this synapses are going to perform well and remain performing well. So that is pruning. Another place where things get connected is the HPA axis. Now, what is the HPA axis? Is the hypothalamo-pituitary adrenal axis. So it is not, how would we put it, a fixed structure. There are various components. One is, of course, the nervous component. So hypothalamus, pituitary, so all the trophic hormones, releasing hormones, and then ACTH, which is the final hormone. So this will also keep on functioning. Okay. Pathogens can, now we saw in that peripheral immunity that there are cytokines. Now, cytokines are chemicals. Many of them will enter through the blood-brain barrier, okay, after a pathogenic exposure, and then they can kind of fiddle around with the hypothalamic functioning. So cytokines stimulate hypothalamus more than necessary. I mean, to an extent it is needed because the HPA axis, the first thing that it will do is kind of ready the body for response, not only immunity wise, but resources wise. Okay. And the medium by which it does is ACTH. Adrenal glands get activated and we have got all kinds of things, glucocorticoids, mineralocorticoids, and so on and so forth. Okay. This is how, whenever there is a challenge, the body gets ready. But what it means biologically is that a peripheral immune or a inflammatory process can have access to the whole system, which integrates the brain as well as peripheral organs. A more simpler, less colorful, less crowded diagram is this. It's the same thing. Whatever happens, peripheral inflammation, which is happening in the blood, in the gut, at different points in life. Okay. The blood, whatever these cytokines or cells are there, they might cross over to the brain tissue via the blood-brain barrier. Okay. Or via the lymphatic system. Whatever is happening, let's say in the gut, will pass on messages through the vagus, through the nucleus of tractus solitarius, brainstem, and then up to the brain. This is how the connections are there. Now, even the lymphoid tissue. So here we talked of inflammation reaching the brain. But is there a connection between the nervous system and the lymphatic tissue? So yes, we have got sympathetic nervous system. Okay. And we also have vagal nervous or the autonomic nervous system, the parasympathetic part. So we have dual innervation, sympathetic as well as parasympathetic. Sympathetic will drive. The parasympathetic, like everywhere else, will break. So sympathetic and parasympathetic, infection stimulates the sympathetic nervous system. How? The next slide, we'll see. So infection stimulates the sympathetic nervous system, which in turn recruits more immune activation. So this process goes on and on. Parasympathetic activity via the vagus nerve helps to both initiate sympathetic activation and later return immune function to normal. So this is a feedback loop. This is how it goes. Pathogens stimulate, cytokines stimulate. Okay. Cytokines will also stimulate the vagal. So basically this is one way. So this is to activate and deploy. So when we look at it carefully, the sympathetic nervous system doesn't we feel charged up whenever we say, no, there's a adrenaline flow. This is the sympathetic activity which is working. So we feel charged, we feel energetic, heart rate is fast. Okay, we are getting ready for the response. So it is a physical presentation, but at a basic level, this is what is happening. We have got more cytokines, which is stimulating the nervous system. Sympathetic overactivity again stimulates this. So this goes on and on till the challenge is met. But then this also activates the vagus, because there has to be something which is going to counter whatever is going on. If the balance is not proper, homeostasis will get affected. So vagus will activate and inhibit the sympathetic. And that is how the loop is formed. So here we have is the same thing. Another thing is, so that was the neural connectivity. So that was the autonomic nervous system, both components, sympathetic and parasympathetic. But all this, whether it is neurotransmitters or hormones or peptides, small proteins, all this have got receptors on immune cells. So these are not found only on neurons, but they are found in each and every cell of the body. And for our interest today, they are found on all cells which are participating in the immune function. So whether it is innate immunity, so we have all those macrophages and all the varieties, microglia, whether it's acquired immunity and we have those B and T cells, all these receptors are expressed on all these cells, and thereby the activity of neurotransmitters, hormones, and peptides will affect the immune activity. For example, which we saw before, norepinephrine binding to beta receptors activates immune cells. So whenever we give beta blockers, it not only gets the heart rate down, but it also can check the immune response or the immune activity. So one of the strategies for cancers is give beta blockers so that immune activity is a bit less. So it gives space for other chemotherapeutic agents to work. Again, serotonin is the same thing. Tumor necrosis factor and interleukin-1 increase the expression and function of synaptic reuptake of serotonin via stimulation of the P38 MAPK pathway. Now, what does this mean? Whenever there is a synaptic reuptake which is high, then we are going to have low levels of serotonin and norepinephrine in the brain, which will produce illness behaviors. And if it goes on and on, it will produce full-fledged symptoms of depression. So if TNF is more, okay, we need to get it down. So newer strategies of managing depression, especially resistant ones, is use something which will get down the levels of tumor necrosis factor. So again, this is the whole list of what happens, dopaminergic receptors on various cells and associations with different kinds of cytokines. Okay, so we looked at the basics of immunity. We looked at the brain immunity interface. Now look at certain clinical entities. One is stress. So we looked at that HPA axis function, and now we have an HPA axis where there is a balance between pro and inflammatory states. This is natural. Homeostasis is maintained. Whenever there is chronic stress, the HPA function, especially in disorders like depression and anything which is tightly stressor related means in terms of anxiety disorders, PTSD, okay, is excessive glucocorticoid activity, which makes the whole system pro-inflammatory. So there is more toll-like receptor pathways, inflammasome activation, okay. So the cells of the body, the cells of the brain are in a hyper inflammatory kind of a state, which we call as a hypercortisolemia. So that is the shift from normal HPA function to abnormal HPA function in depression and other disorders. Now, exposure to early trauma in childhood. What it is going to do is that from very early on in life, the setting of the HPA axis shifts to a hypercortisolemic state. That is one, because there is a constant exposure to stressful situations, whatever it is, physical, verbal, emotional, any kind of stress. If the individual is getting affected by chronic stress, then there are going to be chronic changes in HPA function to a more pro-inflammatory state, chronic inflammatory state. This is what goes on. And finally, this leads to a lot of things. Glucocorticoid resistance. Anything that works, anything that is excessive will stop responding as well. So we have resistance. Gut dysbiosis and so many other things. So obesity, substance use, sleep, other psychopathology. So ongoing stress, continuous stress shifts the HPA function because there's a load on the HPA function to hyperglucocorticoid or hypercortisolemic states. Associated with that, since there is HPA reactivity, there are other changes also. Why? Because we have a hyper inflammatory state going on. So everything in the brain is also hyperinflammation. So we have got everything, whatever we talked of. We will have more microglial priming. So microglia will be always in a phagocytic kind of a predominant stage. Abnormality is glutamate metabolism. We talked of serotonin and the dopamine, norepinephrine functions. Everything gets involved, and then we have either a continuous ongoing experience of stress, anxiety, illness behaviors, depression, and other psychopathology. One of the most studied disorders, which I'm going to brush through because we are not talking of depression. So about the next 10 minutes or so, and then we'll finish. The most variety of findings, number of findings are there when it comes to depression. Okay. So one is the evolutionary pathway. So when we were hunter-gatherers, the immunity was supposed to take care of wound healing and fighting infection because we are constantly interacting with all these things in our environment. And the importance was to manage, so first of all, to become aware of the stressful situation and manage it well. And if there's injury, that gets covered. So there was olden times, increased infectious mortality, decreased inflammatory conditions, because all the time, the inflammatory apparatus, the immune apparatus was managing all these things. And so there was a very less incidence of autoimmunity because the resources were gathered toward the external threats. So these old friends were minimally pathologic. So we talked of tumor growth factor, interleukin 10, and all these kinds of things. Okay, and we were in a kind of an immunoregulatory state, regulated state. Now, when the infections are gone, okay, the inflammatory conditions will arise because now the resources are going to pick up small things which may not be in order. No human being is perfect. No system in any human being is perfect. So there are always going to be some threads, loose ends, something which will trigger autoimmunity. Plus, the stressors are different. They are no longer infections. There are different kinds of stress. So there is a constant stress. So the immune function has changed. Over time, the HPA axis is shifting from okay to hypercortisolemic. So this is one of the evolutionary mechanism of depression. And then there are the different ones. Here we have monoamine metabolism, which we talked of. We also have glutamate, okay, and all those cytokines. Now, what they are going to do is on the crucial areas in the brain. Let's say we have the ventromedial prefrontal cortex, subgenual anterior cingulate cortex, dorsal anterior cingulate cortex, insula. Now, these are the areas which will kind of receive, integrate information coming from the senses, the body systems, with the emotions. The whole experience will get analyzed. So this thing, if it is in an hyperinflamed state, an overinflamed state, can lead to depression. So this is the immunological basis or the neuroimmune basis of depression. So we have got elevated levels of pro and anti-inflammatory cytokines, all of them, but the consistent associations are with CRP, interleukin 6, 1, and tumor necrosis factor. Now, depending on the interleukins or cytokines, we have got different kinds of presenting features. So we had mentioned TNF. So if we want to reduce TNF, we have infliximab. It's a monoclonal antibody against tumor necrosis factor. But when this is more, it is going to reduce serotonin function. So this is one strategy which has been tried. Interleukin 6, if we are able to measure, may predict a good response to ketamine. Baseline levels of interleukin 8 predicted treatment response to a first course of ECT in females. These are study findings. Again, small ones, not very well repeated. CRP more than 1 responded more favorably to tricyclics like nortriptyline rather than SSRI. Interleukin 1 associated with non-remission after 6 weeks. So if we are able to measure, then the responses can be, treatments can be more tailored to get a desired response. Kynurenine pathway will decide probably the outcome of ketamine therapy for depression. So if the kynurenine pathway is primarily involved, ketamine may be the treatment of choice. Now, how do we measure it? We cannot measure it in the lab. These are research findings. So based on the immune findings, immunological findings, we have NSAIDs, which will generally block inflammation. Infliximab, basically for tumor necrosis factor. Ketamine reduces IDO activation, which is the precursor for kynurenine pathway. It stimulated mTOR pathway, decreases other interleukins, but basically it reduces this. That's why it's a marker. Minocycline, also the same thing. Now, not only that, our medications also have an effect on cytokines. So whatever be the medication that we think of, SSRIs, SNRIs, ketamine, TCAs, mood stabilizers, have got different kinds of activities at different cytokine levels or on different cytokines. So patients get better not only because let's say the serotonin function is improved, but at a more basic level, there is a change in cytokine and immune functioning. Psychosis, we are going to encounter the same kind of pictures again and again because the pathways remain similar. Okay, but here, apart from all this mandatory pro-inflammatory, anti-inflammatory imbalances, different kinds of processes which are going on, there is the problem is this altered synaptic pruning. All these neuroimmune changes lead to faulty pruning, which is one basic pathology in schizophrenia, which leads to all kinds of things because we have got circuits which are not optimally functioning. So there is an abnormal presentation in forms of positive symptoms, cognitive symptoms, negative symptoms, neurodegeneration, so on, but also altered neurodevelopment because this thing happens at crucial points in life. Again, the same kind of things when we look at the immune picture of psychosis or schizophrenia, NSAIDs have been tried, minocyclines, essential fatty acids like omega-3, estrogens, and acetylcysteine. The new ones, pioglitazone, which is an anti-diabetic agent, and in some patients, statin have also got potent anti-inflammatory effects. So many physicians and doctors say that statins are the new aspirin. So if anybody is taking statins, please continue. Much more safer than NSAIDs and are known to be potentially anti-inflammatory in nature and hence neuroprotective and many other system protective. Bipolar disorder, again, the areas of interest, frontal cortex, anterior cingulate, CSF, amygdala and hippocampus, so many kind of inflammatory processes are going on. This is just a kind of an atlas. Autism, the whole list, and this list continues to kind of keep on increasing. So we have interleukin 1 beta 6, 12, tumor necrosis factor alpha, interferon gamma, tumor growth factor beta 1, BDNF. There are so many things that have been studied. Okay, again, it's the same thing. Whatever it is an imbalanced state of increased neuroinflammation, abnormal neural and glial responses, and altered brain connectivity. The same thing or a similar thing that we saw in the pathophysiology of schizophrenia. Okay. Yeah, again, this is the same thing. Specific pruning of D1 receptors. If this is too much, this can lead to, as an individual develops, as an individual grows older, socialization deficits. So again, one more pathology, not specific for autism, but even in negative symptoms of schizophrenia that we see. Other disorders, involvement of glutamate in the inflammatory processes by interferon gamma. okay, can lead to neuronal function and degeneration. This is supposed to be one of the processes behind neurodegenerative disorders like Alzheimer's dementia. Okay, this is what it is. Now, and apart from neuropsychiatric illnesses, inflammation, as I mentioned, this is again coming from the first slide, but a little bit more descriptive. In cardiovascular disease, cancers, diabetes, HIV, one thing which I have not mentioned here and which we are seeing in the recent last 5 years is COVID related. It's the same thing. COVID is a virus. It was a new virus for many. Some of us only had a flu-like thing. Some of us had nothing. Some of us had severe illnesses, and many of us died because of complications. Now, why this whole spectrum of things? All of us have the same immune system, but all our immune system do not function in the same way. Because those who had more comorbidity, now let's say somebody had cardiovascular disease right from the beginning. So basically, the inflammation levels are pretty high to start with. I'm overweight or obese, so there is a baseline increased inflammatory functioning going on. We have a challenge, a viral challenge. So everything is going to be now going to kind of manage that. But there is so much inflammation going on, maybe that is not handled well. So that then the virus has a free entry into lungs, anywhere else, and can cause its problems. What are the factors affecting? This is the last couple of slides. Now, again, immunity or immune function or neuroimmune function is not steady. The activity fluctuates over time. So when we are born, let's say this is when we are born, and this is basically the lifespan. Now, as we grow older, okay, the pattern of neuroinflammation changes. This is what it is trying to say. This is the developmental picture. But what happens is that as we become older, what was more targeted toward development now becomes the cause for neurodegeneration. It's the same process, but things have changed. So one effect is aging. As we age, our inflammatory processes, the response is less. It is not very optimal. Second is the level of nutrition, activity levels. Now, one is not in our hands. We all age. 2 and 3 are in our hands. So if nutrition is adequate, activity levels are adequate, they'll provide an optimal challenge, provide resources, provide ingredients for good immune functioning. Not that they are the beginning and the end of it, but they will add to it. Obesity or overweight, this has the number of obese individuals, forget obese, but the number of overweight individuals has kept on increasing, let's say in the last 50 years, especially now in the last 2 decades, especially in our country, especially in developing countries. So we are having heavier and heavier individuals as time passes. So, this is a hotbed. Already things are in place. What things are in place? The inflammation is in place. Microbiome status. Microbiome status depends on, again, these things: aging, nutrition, activity levels, and sleep states. Now, all this together will, at any given point in time, decide how the immune function is going to work well, not work well, predispose us to illness, worsen our illnesses, worsen our treatment responses to illnesses, and so on. So thank you so much, and over to you, Sarika.
Thank you, sir. Thank you for wonderful presentation. And like I said, the complex terms have been simplified by you for the use in day-to-day clinical practice. So there are two questions, or one is rather expert comments from you are expected. Some insight into the role of BDNF. That is the first one.
So, well, we are not talking of growth factors now. The growth factors are something different. Basically, we have got 2 or 3 types of nerve factors. One is brain derived, one is glial derived. So one is, we have BDNF, we have glial derived, that is GDNF. Then we have CDNF, cilia derived. So different kinds of growth factors. Now, growth factors, the prime job of those is to provide a an environment for the growth, the development, maintenance of synaptic structures, so related to the nervous system. They will also get affected when an immune process is going on. It's not that they are immune. Point is that we are not discussing that today. We will discuss when we talk of some more basic science down the line.
Okay, thank you for that. The other question is there any correlation between higher CRP levels and severe or
Yes.
treatment-resistant
Yes.
depression?
So what happens is now getting a CRP is very easy. You can just order a regular routine, that whole list of blood tests which everybody gets done once a year, and we'll get CRP levels. And why is it important is that there is a subset of patients, I would not say all. There will be patients who have got, there will be people who will have on their routine tests a little bit higher level of CRP, but they do not have any symptoms of depression. But that doesn't mean that they do not have any other thing. Many of them will be overweight, many of them will be borderline, early T2DM, mild to moderate hypertension, and so on. So they do not have depression. There will be many people, we do not regularly do blood tests or at intake. When we start treating, we do not do all kinds of tests before starting anything. But if we do, we'll have many people who will have an normal CRP, who will have an abnormal CRP. Now, what it means is that for this person, the ongoing chronic inflammation is in place. Now, what this would mean is, now that is a very broad term. CRP is one of the markers. We do not have all these cytokine assays and things like that available to us on a regular OPD basis, which you can do in patients. So what will happen is that when something like this there, we'll take it as an understanding that it's an inflammatory marker. And with this, we must be kind of prepared that this patient may not respond as well as I would like the patient to. So it is not going to tell me what is going to work well. We can try tricyclics. They do take care of this kind of things better than probably SSRIs. But that strategy is not 100% solid. Only thing is, yes, that an inflammation is in place. Yes, whenever there is baseline inflammation, there can be a lot of comorbidity, including other physical comorbidity. And lastly, well, it might be not taxing for our thinking as to what if this patient, whether the patient is going to respond well, not respond well, what other things are needed. So yes, there is a possibility.
Yeah, so to continue with this, let's say I have a patient whose baseline CRP is higher and I started on tricyclic. When should I repeat the CRP? Is it 6 months, 3 months, 1 year, or do we see any
No,
change? We
see
don't see
this
any
kind
change.
of markers, CRP, it doesn't take very long for it to kind of come back. But let's say 3 months is a good deal.
Okay.
If
So,
I do it next week, may not be really. We need to give some time.
yeah, so with our medication, I think 3
Yes,
months is fair enough time.
because I think at least a month and a half will require, especially if you're using SSRIs, to build up a dose.
Yes.
And slowly we'll see the effect. So what we have to then correlate is the improvement in depression with the improvement in CRP.
That's right.
But
There
it's
is
an
one
easy exercise to do because CRP is easily available. Anywhere it can be
yes.
done.
There's one more question. Please throw some light on SLE and psychiatric symptoms.
So we have SLE as a multi-systemic illness. And we have those typical antibodies that are formed to surface antigens. So it's an autoimmune disorder. So this comes under the purview of what I talked about, sterile inflammation. This is not driven by anything which is an external challenge in terms of any organism or a pathogen. This is happening because my cells are now kind of showing certain marks which are picked up by my, now over a long period of time, the acquired immunity. So cells are getting damaged. Now this SLE actually can happen anywhere, whether it is skin, whether it is kidney, whether it is brain, anything. Okay. So when we have this anti-DNA, that group of antibodies, we know that there is a lupus going on, and it can actually present with any kind of a symptom. In fact, many a times the presentations may be initially psychiatric. Now the problem with us is that when somebody comes to me with depression symptoms or some psychosis, something, we start treating. All this what we learn in terms of theory, we keep it aside. We are happy that the psychosis improves, depression improves, and then our work is done. But for many illnesses, psychiatric symptoms can be the presenting complaint. We get into this kind of an understanding only over a period of time. Again, for every patient who comes to me with depression, whatever age group, male, female, okay, with whatever other kind of, other kind of comorbidity apart from SLE, we start treatment. And even in those patients, we do not get this kind of an anti-nuclear antibody kind of profile done. So what happens is we don't know. But what we will then experience is people not getting well. Some other funny kinds of symptoms will come up. Somebody will come with some kind of rash. We say it's drug induced. It may be something else. Okay. So, but yes, SLE is very common. It's not that uncommon. So we need to be aware that this can happen. Now, again, when we have these medications, what we talked about, our medications having a kind of a immunomodulatory role. When we use them wisely, we can also have some kind of an effect on the basic pathogenesis of SLE. In everyone, no idea. Potentially, yes. I hope that answers the question.
Yes. Rajkumar sir, anything you would like to add or say?
Yeah. Thank you, Dr. Sarika. Thank you, Dr. Malabai. Thank you, friends. And with this, we end up today's session. Next session is going to be personalized medicine in psychiatry on the 23rd of this month. Till then, good night and goodbye to all of you.
Good night.
Thank you, sir. Thank you
Bye.
for the opportunity. Thanks.
Bye.